Hi, my name is Justin Wagner, and I am a computer scientist at NIST, and I work on the Human Genomics team. So before I get started with my presentation, I'd like to thank the organizers for the opportunity to present this talk. Today, I'll be presenting work in the Genome in a Bottle Consortium that is working to develop a comprehensive variation benchmark for medically relevant genes on chromosomes 1 through 22. So a little background on the Genome in a Bottle project and the benchmarks that are generated by it. So NIST co-leads the consortium, and the Genome in a Bottle Consortium develops metrology infrastructure for the characterization of whole human genome variant detection. So the way NIST has accomplished this is that it helps develop 7 broadly consented human genome cell lines, including 2 mother-father-son trios shown on the right-hand side of this figure. And so from one large growth of each of these cell lines, the DNA was extracted, and the extracted DNA was actually turned into what's known as a NIST reference material. So reference materials include reference values, and so the reference values for each one of these reference materials are the whole human genome germline variants that are detected and can be used to benchmark sequencing technologies and variant calling methods. So the evolution of the Genome in a Bottle benchmarks. So this project started about 2012. And so the first 2 years of the project, there was focus on the pilot genome, which is NA12878, or in the Genome in a Bottle labeling HGO1. So the work developed a small variant benchmark on GRCH37, and this covered about 77% of the reference. And so this allowed methods developers and sequencing technology developers to characterize how well their variant calling methods and variant detection was performing on a specific reference material. So from 2015 to 2019, there was work expanding the number of samples in the Genome in a Bottle collection to include members of different ancestries, and then also focus on developing trio samples, so mother, father, son. So during this time, the Genome in a Bottle Consortium published a small variant benchmark that covered between 80 and 90% of GRCH37 and GRCH38 for these 7 samples. And then also, for better use of the benchmark variant calls, Genome in a Bottle worked with the Global Alliance for Genomics and Health to develop a set of best practices for comparing a variant call set to the Genome in a Bottle benchmarks to identify performance. So recently, Genome in a Bottle has also published structural variant benchmarks and also a phased diploid assembly of the Major Histocompatibility Complex of HGO2, and also developed with long read and linked read technology a new version of the small variant benchmark that covers more challenging regions of the genome and covers between 92% and 94%, depending on GRCH38 or GRCH37, of the human genome on chromosomes 1 through 22. So I'm just going to briefly walk through our recommendations for how to use the Genome in a Bottle benchmarks. So the benchmarks, the small variant and structural variant VCF and bed files containing the regions of the genome that include those variants, are available on the FTP at the bottom right-hand corner of this slide. It's hosted by collaborators at the NCBI. So that FTP also includes, beyond just the released VCF and bed files that are used as benchmarks, it also includes sequencing data that's publicly available for each one of the cell lines, both short read and long read sequencing, as well as different analysis that has been done by the Genome and Bottle Consortium, so different variant call sets, and then draft benchmarks. So the releases folder that I denote at the bottom of the slide, so that contains the VCF and benchmark beds that have been evaluated against a variety of call sets. And so the resource development process that we go through is detailed in manuscripts and then also preprints from Genome in a Bottle. Another important item for identifying performance of variant calling methods is stratification of the human genome, and there are stratification files that have been generated by Genome in a Bottle that are available on the FTP. And an example use case in a recent Precision FDA challenge in which we released the version 4 benchmark and sequencing data for the HGO2 sample, and then released just sequencing data for the parents of HGO2. Then people would submit the variant calls on HG3 and 4, and we compared them to benchmarks that we had generated. And so a manuscript describing both the stratifications and then also the performance of these different methods is at the link provided. So on the right-hand side, there's a figure from the best practices benchmarking paper. So what any user we expect to have is a VCF that contains the variants from any of the Genome in a Bottle cell lines. So what we provide is both overall truth VCF, and then also a bed file that contains those variant, the regions that contain those variant calls. Then also we make available the stratification beds. We recommend using a tool like hap.py that both wraps comparison tools. We commonly use the comparison tool VCF eval to compare two VCFs, and then quantify the performance in the different confident regions and stratifications using hap.py, and then a report detailing performance across the different regions of the genome between the given query VCF and the ground truth data set is provided for users to understand how well their variant calling method is performing. So in terms of new benchmarks, we recently generated version 4 small variant benchmark. And so this used PacBio Hi-Fi long reads and then 10x genomic linked reads. So this was an improvement over the previous version, the version 3 of the benchmark, in terms of coverage of the non-gap reference genome. And then also we included the benchmark that was generated using the phased diploid assembly of the PacBio Hi-Fi long reads of the MHC. And so you can see that the coverage has increased for version 4 compared to the previous benchmark version. And then also the coverage of low mappability regions and segment typifications has increased substantially compared to version 3. So version 3 was largely based on short reads, and then version 4 included the short read call sets as well as the PacBio Hi-Fi long reads and the 10x genomic linked reads. So this gave access to more difficult regions of the genome. And so overall, you can see for GRCH37 and GRCH38 from the table below, version 4 added about 400,000 single nucleotide variants and indels less than 50 base pairs to the benchmark data sets. So looking at medical gene coverage of version 4, we have a figure from the manuscript on the right. So we wanted to take a look at how well version 4 with the long reads and linked reads were covering what were known as medical genes that were difficult to characterize with short reads. So the criteria for that was that Mandekler et al. In 2016, they created a list of medical genes that had at least one exon, which was difficult to map with short reads. So for the version 4 manuscript, we looked at how well we were covering these genes. So this figure shows that compared to version 3, there's a marked increase in coverage of these genes. But ultimately, we don't cover all medical genes that were difficult to map with at least one short read. So the project I'm going to describe today goes through trying to generate a genome in a bottle benchmark for as many medical genes as possible. So including the criteria that was given from Mandekler et al. Of at least one exon being difficult to map with short reads. We also wanted to look at how well we're doing across all medical genes, or a set of medical genes that we could find in literature. So overall, looking at how well version 4 on GRCH38 covered the set of medically relevant genes that we found on chromosome 1 through 22, we discovered that there's 354 that were less than 90% covered. And so the reasons included there were structural variants that we exclude from the benchmark. There were large segmental duplications where it was difficult to characterize. And then also other difficult regions that even a mapping-based approach with the long reads weren't able to resolve. So in this project, we looked at using a diploid assembly. So there was the Human Pangenome Reference Consortium in the spring of 2020 put on a bake-off of different diploid assembly methods for the HGO2 sample. And we used the best performing diploid assembly to help us generate a phased small variant and structural variant benchmark of as many of these medical genes as possible that weren't fully covered in version 4. So we ended up generating a benchmark for 273 medical genes, and that's what I'll be detailing today. So this figure gives an overall view of how we generated a medical gene benchmark. So I'll go on the next set of slides in depth to several of these processes, but for the high-level view, we started off with finding a set of medical genes. So then we used a trio-based diploid assembly from a tool known HiFi-ASM, and we looked at how well it resolved each one of these medical genes compared to version 4 benchmark. And then we went through a process of manual curation and comparing to different variant call sets to generate a benchmark. And then we have gone through the benchmark evaluation with the community, and we're getting ready to release this benchmark for use. So first, looking at the medical list of genes. The first large set of genes that we worked with was from the Mandekler et al. Paper. In addition to the focused set that were at least one exon were difficult to map with short reads, they also just gathered a list of all medical genes at the time. So this was a snapshot of OMIM, HGMD, and ClinVar. And the snapshots were taken between 2012 and 2015. And so this resulted in 4,773 genes. We also took a look at a product from the COSMIC project, and this was the cancer gene census, and that included 723 genes. Then finally, we looked at a set of genes that are commonly sequenced by clinical labs. So this was a collection of a union of the ClinGen definitive, strong, and moderate evidence genes, the ACMG secondary findings 2.0 list, as well as other carrier screening and commonly tested in clinical lab gene lists. So the figure on the right just details the intersection of these different gene lists. So the diploid assembly that we used to generate this benchmark was FactBio HiFi long reads. So the method to develop the assembly was HiFi-ASM, and we used the trio information was used. So this meant that the Illumina reads for the parents, and then the PacBio Hi-Fi reads for the son of the AJ trio were used to generate the assembly. So this was the best performer in the Human Pangenome Reference Consortium Diploid Assembly Bake-Off in spring of 2020. So the figure on the right is from the Hi-Fi SM paper. So the Hi-Fi reads are error corrected and assembled, and so this results in two haplotypes. And then we took the haplotype assemblies, we aligned them back to each one of the references, so GRCH37 and GRCH38, and then we called variants with a tool known as dipcall. So this outputs variant calls as well as confident regions. And so the confident regions for dipcall is that each one of the locations is covered by exactly one contig from each haplotype. So we use the dipcall confident regions to examine genes that were less than 90% covered from the version 4 benchmark. And we added a criteria for the dipcall confident region coverage of each gene, which included flanking sequence that was equal to the PacBio Hi-Fi long read length, as well as being able to fully resolve any overlapping segmental duplications of the gene. So this resulted in 273 genes that we then curated each gene for the accuracy of resolving the assembly in IGV. And after we had this draft set of genes and benchmark variants for them, we then requested call sets from the community. We compared call sets to these draft benchmarks, and we manually curated a subset of false positives and false negatives from each call set. And so the call sets are listed below. Those include PacBio and Illumina-based call sets, as well as ONT, and called with a variety of different calling methods for both small variants and structural variants. So looking at overall statistics of the medical gene benchmark within these 273 genes, so compared to the version 4 benchmark, it includes more bases, more SNVs, more indels, and then importantly, it includes more coverage of the low mappability and segmented duplications within these gene regions. So there's also a SV benchmark that we've generated, and so that included 217 large insertions and deletions. To test the utility of this benchmark, we compared an Illumina NovaSeq GATK4 call set against the version 4 benchmark in these genes, and also this medically relevant gene benchmark. So overall, the medical gene benchmark detects more errors in segmental duplications and tandem repeats, and then also fewer variants are excluded by the new benchmark as compared to the previous version 4 benchmark within these genes. So highlighting a few genes in the new benchmark include NCF1, which is within a challenging segmental duplication, and the gene is seen in a subset of patients for a primary immunodeficiency disease. So this provides us, this assembly-based approach provides us an improvement over the alignment-based methods that we have used historically in Genome in a Bottle. There's also another gene, which is shown at the right, which is CBS, which is involved in homocystinuria. And so CBS is interesting because the medical gene benchmark properly resolves this gene. Looking closer at this, what the issue is with mapping-based approaches is that in the GRCH38 reference, that there's an extra copy of this gene, as well as a set of genes on chromosome 21. There'll be a blog from the Genome Reference Consortium detailing how to handle this approach, as this is how to handle addressing this issue in GRCH38, and this has been raised by the community. And then also, the diploid assembly properly resolves the gene SMN1. So SMN1 and SMN2 reside on chromosome 5 in a large segment of duplication. It has historically been difficult to sequence, and SMN1 is involved with SMA. And so with the diploid assembly, we were able to resolve both haplotypes of SMN1 for the HGO2 sample. And so this new benchmark allows for characterizing performance in this challenging region. So we don't cover SMN2 yet, but we will be looking at future diploid assemblies to identify if this gene is, if SMN2 and SMN1 are both properly covered, and we can generate a benchmark for that. So the overall conclusions I want to leave you with are that the version 4 small variant benchmark from Genome in a Bottle used long and linked reads to enable benchmarking in more segmented duplications and low mappability regions. So this improved overall coverage of the genome. There were still medically relevant genes that were not in the version 4 benchmark or not fully covered by version 4. So we wanted to explore ways to expand our coverage for clinical laboratory stakeholders. So we looked at using an assembly-based approach for developing a benchmark. To do so, we used the TRIO Hi-Fi ASM, and this allowed us to benchmark in these more challenging medical genes. And this allowed us also to do methods development to generate future benchmarks. So the overall result is that we have a phased small variant and structural variant VCF embed that will be placed on the Genome in a Bottle FTP that can be used for benchmarking these medical genes in the HGO2 sample. Future work we are undertaking in Genome in a Bottle is to produce a chromosome X benchmark, because many of these medical genes are on the chromosome X, as well as a whole genome diploid assembly-based benchmark. So I'd like to thank all the Genome in a Bottle contributors and collaborators on this work. And so this involves many stakeholders in the community. And I'd also like to thank the organizers again for the opportunity to present this talk. And I'd be happy to answer any questions about this work.